Do I want to have extra cheese?
Oh, I got to go on a nice little run.
Where'd you go?
The team ran to the girls
Oh, nice.
from Ballard.
Does everybody have enough cards?
It was a good run.
Yeah, just about. The only downside is raining.
Are you gonna do it?
Possibly.
I'm trying to get my friends to sign up, so I can really start running.
It's so scary.
Yeah, the only downside is it was raining. Not for most of it. Most of it's like a straight line because it's just Ballard there. But then like the last, I'd say probably quarter of a mile, it's like there's 3 big hills that get like progressively steeper, and it's right at the end. So of course everybody's already tired. So that last bit was is a stretch, but so we got there, and it was nice because I had an 8 a.m. So I got to ride back with the coaches. I didn't have to walk back. Some people had to walk back.
3D.
It was fun.
Good way to start the day, I guess.
Let's do it, guys.
The camera's stuck over here. The camera is stuck over here.
Once I start talking, or once I become the loudest one, you should follow me. Hello?
Follow me back to my. It sees your torso.
All right, it's gonna come back.
No, just stop.
We can do it in a few minutes, it'll catch up.
Okay.
Go ahead. Let me open up the screen. We're good. All right. I finished reading everyone's papers, right? The first paper, I should have given everyone comments back on their paper, either on Brightspace or on the actual document directly if you give it to me on Google Drive. If you haven't gotten any feedback on your paper at all, send me an email and send me the link to your Google Drive and I'll try to give you some feedback on it. But first, check out the 2 spots. For that paper, you guys did pretty good. One of the things that I caught on a little bit later was that I forgot to tell you guys to, if you found some kind of weakness in the paper or something you didn't feel comfortable with, you got to make sure you bring it up. Yeah. For one of the papers, they were doing an experiment where they were trying to figure out what bacteria were present in people's mouths, but they only checked 8 different people. And then they were it claims that from 8 people, they can say that all the bite marks were completely different. So yes, you can have those results for that small group of people. Would you let that represent a population? That's why I was kind of like, So if you see something like that in the paper, something doesn't feel right, something's wrong, or something, even if it feels unclear or it's not really well explained, you can bring it up on your thing, right? The other one is, for the most part, I feel everyone had a pretty good understanding of the papers. Like I can see that you guys went, figured out what was the main thing, and you talked about it. There was a few papers where it felt like you read over the paper really fast. And you had like, hey, I glanced at the paper and I was like, this is what I think it means. Because when I looked at the paper itself and then I look at your writing, there's like some really big differences in what they're talking about. So it makes me think like, you might have just read the abstract and you wrote down the paper, and then the abstract and a few papers weren't that nice. So make sure you're reading the paper. It takes time to read them. It takes a little bit of practice learning how to get the information out of these, but they're super important to get that skill. If you feel that there's stuff that somehow it's like you're having trouble getting the data out, make sure you guys come to office hours. Like just come chat with me, bring the paper with you, and I'll go through the paper with you, trying to break things apart. It's a really big skill. You guys have to learn 243, which is like molecular methods. Like literally we spent about a month working on just one paper. Like they take time to break them apart. So make sure when you guys are doing this next one, give it a good read. And then you'll get like a nicer write-up. I know it's over spring break, so I might answer emails. Try to send some stuff out to me. Sometimes I'll get on, I'll be able to send stuff. All right, so that's taken care of, the big stuff. For the next paper, the other thing is gonna be, again, my big thing is no quotes, right? I need you guys to process the data, understand other things, bring the data and present it to me. If you just copy the exact test and put it what they said, I don't see a chance for you to show off what you're understanding or summarize it for me. So make sure you guys take care of that. Next thing, we have the review session on Monday. The first midterm, I went hard on the review material myself, right? But sometimes making that the journey of creating the material, whatever works best for you, is part of the learning process, right? Like you have to be able to grab a bunch of data, figure out what's important, and kind of make a cohesive story out of it. So this review session, I'm not gonna make the material for you guys. So I want you guys to chase it down. I tried showing you as many ways as I could, right? I gave you videos to support the data. I gave you the pictures from the slides. I gave you a list of vocabulary words, and I gave you guys write-ups, right? So I'm hoping that you guys found which study method or which technique helped you out the most. That way, I want you guys to kind of go through the notes, try to pull that information out yourself, you know? On Monday, we're having a review session. So for that one, I'm not bringing any material. So whatever you guys bring to class from your reviews, whatever it's unclear, that's what I'll go over. Yeah. There. Yeah. No, okay. We're gonna try it out, guys. So review Monday, and then on Wednesday, we take the test. I don't think there's a class after us, but if you need extra time, I'm gonna try to keep it reasonable. If you need extra time, we'll probably just take up another room. Just if they kick you out from here, we'll go find you somewhere else. Yeah. And I think that's the big stuff. Pretty good. And then the other one is today, again, that piece of paper. Try to think of stuff that feels unclear, or if you have a question, like you can kind of propose it for the midterm. If I pick your question, I'll give you extra credit points for it. The other one too is like if you help or bring up stuff in the class to make us discuss things, and then it helps the class out, I'll also give you bonuses for that. You know, right? So I really like making sure you guys do that, because normally that's the kind of stuff people write on their letters of rec. When they're like, oh, they participate in class, the discussion point, and they bring up the level of the conversation that we're having. So even if you don't want to do it here, do it in your other classes, get more people to notice that about you. Yeah. And I'll try to reward you with points. Good to go?
So we're just writing down like ideas for the
It's up,
midterm?
yeah, it's up to you. It's either stuff's unclear, like the question when we're done, you're like, hey, I don't get this, write down the thing that you understand. The other side of that is if you see how the material is kind of set up and you're like, oh, I think I can write a question for the midterm with using this material, just write it that way. And then if I use them, I'll give you points for it. You know? So put your name on it. All right, let's go.
Do the cards that we did last time, are they, the extra credit gonna go on the next exam?
No,
Or
it's
like
on this one.
This one, yeah.
Because these are for about just the material on section 2.
Okay.
So section 1 material, I'm gonna try my best to not go back to it.
So is it gonna be not necessarily cumulative, other than like knowing the general idea of things, but more specifics of this?
Yeah, perfect example would be like about today. It's for the section 1, I went over super heavy into how the enhancers bind by activators and they lead to activation, and then I go down the whole downstream thing with polymerase, blah, 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 right? Today I'm gonna show you, I'm just literally gonna stick up here where I'm talking about how is it that we set up the histones, how do we take them off, like what are the mechanisms, and also how do we, when we're copying DNA, we keep those marks on, right? So activation as a general idea, all I'm gonna hit for transcriptional regulation is transcriptional activators, repressors. I don't care anything downstream.
Okay.
Yeah, but just focus on the top part.
Also, is the exam still gonna be in person despite Mr. or Dr. World's being online? Cause I think
I've been
theirs is,
thinking about switching it.
theirs is online and after spring break. I know.
It's
The thing for me is like, when I do online tests, I normally don't do, like, what does this mean? What does that mean?
Mhm.
I normally switch those over to being more problem solving, right? So it's like, I have to write a different type of test. I've kind of thought about it, and I'm like, I don't know if you guys are gonna be okay with me, because it makes the questions a little bit harder to tackle, because it'll be like, you have to know the foundations all by yourself, and then I'm gonna probe you on how to break things, and you have to figure out what should happen. I think maybe not this midterm. We can talk about it for the next one, and what I'll do is I'll switch over my questions for the quizzes before class, and then you guys see if you are okay with it.
Just another piece of information. Dr. World's tester also he gives 6 questions out and he lets you choose 4 of the 6 to grade.
And I think that his are explaining pathways or how things.
I think, yeah, it's
kind
more
of a
problem
series of
solving
things
too.
happen.
I think.
Yeah, that's the thing. So it's like I have to switch. Like right now,
Yeah,
like the first midterm, right? Like that first page is literally definitions, right? So it's like that's level 1. Should be the easy stuff. You know the word, match it up to what your world goes with. like in that, like I said, in that scenario, is like, I would have to switch you guys to problem solving. And then it's like, it's more challenging, and you technically have more time to do it, but that's because it's gonna be more challenging. I mean, I'm a little hesitant to go there, because I get too excited about those kind of questions, and then I'm gonna make them really challenging. That's what I'm scared of, right? But I have a blast. I'll do it. So I'll give you a few practice rounds, see what you guys think, and then the next two midterms you guys want, we'll switch over to doing problem solving. Yeah. All right, let's get started for today. Any other questions? No? All right, today's gonna be a fast day. Again, we're really gonna be looking at just the DNA itself. So for this, we're gonna think about first, it's gonna be just Mendelian genetics, right? So for this one, you're thinking about in the DNA itself, you have, let's say again, we're gonna go with a little bronze. The chromosome, somewhere in your chromosome, you're gonna have one of those critical genes, right? If, let's say your parents have different versions, this could be the chromosome, and you'll find the other gene that you got from your other parent, right? So let's just say, this is my mom, this would be her genes. Let's say she was homozygous, has 2 copies of the exact same gene. My dad, we put him as heterozygous, right? In this case with him, let's say he has 1 bad copy, 1 good copy, right? Homozygous, she has 2 of the good copies. And when they pass down things to me, I'm still heterozygous. I got 1 good copy from my mom, 1 bad copy from my dad, and I have this, right? Based on what you have, you can then determine what kind of phenotypes you'll get, right? So in this case, they're kind of showing you for plants different things that can happen. So it'll be like the shape of the seeds. This could be the color of themselves. You can see the effects in the flowers, shapes of the fruit. All these different things are going to be working on the same pathway, right? Depending on how your genes get to the organism, it's going to depend on how the organism is going to work. And then finally, let's say I have a kid, and let's say my wife has this type of mutation, and then you give it to our kid. Now this kid would be homozygous with a mutant version, right? I hate the way that they name this stuff, because it's like, they write it like, they'll say like yellow, and they'll write minus, and then yellow plus. And then this is meant to be like, yellow plus means that you have yellow and it works. Yellow minus means that it's either mutant or something else. They put the minus, right? The mutation for the wild type. It's really confusing because you'll see like yellow minus could mean the plant is not green. But this is the way, what we're looking at, the different mutations, they give us these final outcomes, right? So here, if we drew them as Punnett squares, here's the other way to write it. Instead of doing plus or minus, you can also do it with capital letters and then lowercase letters, right? So in this case, on the left, we're looking at the phenotype. That means it's wild type. Lowercase means it's mutant. When you mix 2 heterozygous parents, you get this really cool distribution. And it's normally that when you look at the genes that you produce, right? There's a chance that a quarter of your population of your offspring are going to have 2 wild type copies. Now you have a homozygous wild type, right? Some cases you'll get 1 good copy, 1 bad copy. Now you're heterozygous. But you'll see there'll be 2 versions of this, right? One where you get the wild type copy from 1 parent, or you get the wild type copy from the other parent. And then finally, you have this, which would be a quarter of your population is going to show up looking like the mutation, which will be this. In this case, we're looking at a mutation that is recessive, right? That means that it only shows up when you have 2 copies of the mutation. These are opposite to the ones that are called dominant, which means that when dominant mutation, even if you have 1 bad copy, the phenotype shows up. So here, genetically, you have 3 species, right? Wild type, 2 heterozygous, and 1 mutant form. Phenotype wise, like what you're seeing, you have 3 things that look wild type and 1 that looks broken. If they're dominant and it's working backwards, what's going to happen is you'll have, again, 3 different genotypes, but your ratios will be 3 of them will look mutant, and only 1 of them will look wild type. Does that make sense? Yeah? All right, let's go genetics. Here on the right, they're kind of showing you that, for the most part, the genes kind of distribute randomly, right? So if you're looking, instead of just 1 gene at a time, now we're following 2. Here we're looking at brown and yellow there's a chance that that brown gene could either lead with the wild type version of yellow or the mutant version of yellow, right? If your parents are heterozygous. At the same time, the wrinkled gene or the mutant form of that, lowercase r, could potentially lead with the yellow or the mutant yellow. Yeah. Same thing, you draw up your pattern, depending on what your parents are, you kind of figure out you have this distribution. And then again, now, because you have two genes that you're following, you'll figure out that you get a lot of different patterns. The thing is, since they distribute by themselves, you can kind of look at it as, if we're just looking at the wrinkled mutation, which is a recessive mutation, you can see that a quarter of our population has it, right? It's 4 wrinkled, the other 12 are not. But because we're looking at 2 different genes, you can see that a quarter of this population, a quarter of this should have the second mutation, which is that recessive thing to get us no yellow, making the plant green, right? So you can either use these tables to figure out what the mutations are. The other one is you can treat them independently and just know that you just have to multiply the 1/4 times 1/4, and you get that 1 in 16 to give us the results. Yeah. So this is the regular rules, and we're saying the genes are coming in, depending on what gene is present, right? Either, in this case, right, my dad looks normal, my mom looks normal, I look normal, then my kid could potentially have the mutation, right? That's if you follow exactly what the genes are there and what they're present. The thing is that we're gonna find out that there's different levels of regulation that actually mess with that math, right? There's different ways that we can actually cloud these things. So here we're gonna talk a little bit about like incomplete dominance. So the trait's trying to be there, but we have different ways to try to regulate it. The other thing that's gonna happen too is here, the yellow color, the shape of the seed, they're single genes. One gene is able to carry out the function. For us, things are a lot more complex, and it takes more genes to set up a single functional thing, like our eye color. I can't remember exactly how many different genes it requires to work together to be able to give us the color. In that case, you have a mutation in one single gene, you really can't change the color of our eye color. You have to hit multiple things to make it happen. And then the final thing we're gonna be talking about is gonna be this thing called epigenetics, which is, even though you could have the DNA, there's different ways for us to turn on or turn off certain pieces, right? So we're gonna get a little bit into that today. So for this one, the big ones we're gonna be talking about, it's gonna be about, I'm not gonna go directly into the genes themselves, but I'll show you different ways that we can actually put away the gene. If you put it away inside the histones, technically your cells behave like you don't have it. So let's get a little bit into the mechanisms. So first thing that we're gonna be able to play with is gonna be at the level of, we can put the DNA away into the chromatin, right? So it's a big complex, wrap it up around the histones, you have this big protein aggregate. By doing so, you basically have whatever gene, let's say this mutation, like just say in my case, if I coil up this piece of the DNA, technically I only have the good copy. The phenotype won't show up, right? Like I'll look wild type. If I accidentally put away this version and only have the mutant version available, despite me being heterozygous, I can still show up with the phenotype. Does that make sense? Yeah. All right. So how is it that we control that chromatin? For this one, I talked a little bit about it. So we have the 2 different states. We have the euchromatid and the heterochromatid, right? Euchromatid is when it's open. That means that the gene should be available. In the heterochromatid state, it's going to be in the closed state, right? So tightly wrapped around it. So for this part is, how do we determine how tightly or how loose the DNA is going to be wrapped, right? And for this part, what you're going to notice is that we have these 4 histones, H2A, H2B, H3, and H4, and they basically have tails. What you're kind of seeing here is this protein sequence. Those tails, they normally have residues that have positive charges, right? Protein has positive charge, your DNA has a big negative charge, they actually attract each other, right? And that's the way that you can get this really nice tight binding. The thing is that you can regulate this by doing different modifications. Like you can covalently bond different things, and you can change the chemistry of those bonds. So for this one, we're going to be talking about 3 different types, but I'm going to focus on the first one, right? So we have acetylation, phosphorylation, and methylation. So acetylation, when we look into that one, we're basically going into that amino acid, we're zooming into the R group, right? This would be this right here. Normally unmodified, there's your amine group, there's your nice positive charge, right? So in this state, perfect, we have the DNA tightly wrapped, we wrap onto it perfectly, it's basically stored away. We bring in these complexes, right? Here we have the histone acetyltransferase, I should highlight that. That's gonna be an enzyme. This is the one where it connects to transcriptional regulation, right? So this would be like your activator. And bring in this enzyme, what it's gonna do is it's gonna help us modify the different tails. It's gonna add this little methyl group, right? Methyl, and then it's gonna be a double, C double bond O. But this is gonna give us negative charge, right? And you basically can't, not negative charge, it's canceling out something that was positive before. Like taking away the charge from it, now you're gonna not have it stick to your DNA that well. So at which point, that enzyme is basically kicking, or loosening the DNA from the histones. So it should uncoil your DNA, or unravel it. Right? Opposite to this is, in a repressive state, your repressors, and when you have protein interaction, they recruit those, the opposite, which is this H stack, the histone deacetylates. These guys will come in, cleave this, get rid of the acetyl group, and now you're back in a positive state. Right? So when you do that, this brings in, it should coil the DNA back up onto itself. So by modifying the activators and repressors, you modify the hats and the H stacks. Those modify the tails, but by modifying the tails, you either have open or closed DNA. That for sure is going to be a quiz question. Yeah, so be comfortable with all. What is it? One. So level 1, the repressors. Level 2 is this H stack hat. Level 3 is what's happening at your histones, and level 4 is how your DNA is going to be in that shape.
So the hat and the HDAC bring in these, like bring in or take out the acetyl group?
Yeah, they're like, so imagine them like the enzymes that are catalyzing the reaction, right? So either they're gluing it on or they're breaking it off. But they will modify the histone tails in their environment. The thing that's gonna put them on the gene that we care about is gonna be these guys. Because remember the activator and repressors, the proteins, they have a DNA binding domain that likes a specific sequence. So when they're present, they'll go find, like let's say this was the gene that they care about, they'll go and they'll bind to the piece of DNA surrounding it, and they'll basically recognize it. Right? That'll be your activator, let's say. This activator then, through physical protein-protein interaction, will recruit the hat. Right? Now that enzyme is being held in place and it's modifying all the histones around it, and that's what's gonna lead to opening up, it's gonna lead to this modification, which ends up opening up the DNA. So yeah, this one, quiz question, for sure. Any other questions on this part?
And you say by opening or closing the DNA, it's like turning it on or off. Is that what you're saying?
Downstream, that's what's gonna happen, right? Because it's gonna be, in this case, like this drawing, to be honest, doesn't do it justice. Like when I used to draw it in my old class, I used to draw it like this, like. Let me see, yeah, let me show you. You'll see this. I normally draw it more cartoony, but the goal is if that's, let's say you're repressed state, normally there I would draw my histones like super compact with each other. Like they could be like maybe 10, 15 bases between each chromosome, I mean, between each one of those nucleosomes kind of attached to each other. So they're super compact. At that point, all your critical stuff, your title box, right? All the places where your transcription is blind, you're going to be completely hidden. And if that means that you won't be able to make that gene at all. That's when the activator comes in. That'll be that first signal to say, hey, this is gonna bring in our hats. Our hats are gonna modify the tails. As they do this, these guys basically start to uncoil from each other. So then you'll have bigger stretches of DNA open from each other. That's when you basically expose those nice binding pockets. Title box, the TBA, TB, and all the transcription elongation factors will be able to set up, turn the gene on. Yeah. Yeah, so probably, again, if I ask you a question about activation and repression, You can get me to here, and I'm okay, right? You don't need to explain the different factors that kind of help express it, just get me to where you can turn it on. Yeah.
On the other side that says histone modifications, and it had all of the like K, the one before. What do we need to know about like the K and S? What are those and what do we need to know about that? I still don't understand.
So here, euchromatin, right?
Mhm.
What does that mean?
It means that it's open and like gonna, you know,
Yes.
make the proteins.
So these three things will be the things that'll set up the euchromatin, right? So
Okay.
the main one I'm gonna go into is gonna be acetylation, just because I want to show you the diagram, right? This thing, how it changes the charge. The second one, I can show, let me show you a quick picture. But it's, since you're bringing a phosphate, you're gonna bring one phosphate, and it's gonna have like 4 oxygens surrounding it. So you're gonna bring in like a super negative charge. Phosphorylation, kinase, phosphatase. I'll try to find you a nice pretty picture that summarizes it faster.
So are those like the genes that acetates or
So so everything we're looking at is happening at the protein level, right? So it's like, if I was a histone and it has your arm out, here's where we can have those different amino acids, right? In the case of acetylation, right? You can imagine, like, let's say at this first slot, which is gonna be your lysines, you're gonna have a positive charge there. The moment you bring your acetyl group, you get rid of the positive things, that'll help release the DNA. The other way you can do that is, you can work with kinases, right? Kinase is gonna bring the phosphate group, it's gonna bring in a big negative charge.
and that's those two things?
That's,
Are those two the kinases?
oh no, so this, let me draw one out real quick. I think it's gonna be best if I draw it. So again, if we're super zoomed in, red's gonna be, green will be my histone, right? So this will be that protein. We're only gonna be looking at one of them. There's the tail, like that, right? Normally has the positive charges like this. This is what helps keep the DNA wrapped with it. It helps with the interaction. So one of the things that can happen is, if we have that tail and one of these positive charges, you now bring in your acetyl group. So acetylation, this is gonna have a negative charge now. Because you brought in a negative charge, you're not gonna be able to hold on to your DNA tightly. This switch, going from this state to this state, ends up happening, right? To go from here, going down, that's your histone acetylase, right? That's your hat. That's gonna add this little molecule right there.
Okay.
Then to get us back from this state going upwards, this is when your H stack takes over, right? So you can switch back and forth. So this glues it, this cuts it.
So is that a positive charge or just a positive factor?
The amino acid on your histone has a positive charge. You're bringing in that double bonded O is the negative charge that cancels it out. So now in that domain, you no longer have a positive charge.
And that's what shuts it off?
That's what determines what state you're gonna find your DNA in, right? going from here to here, because now you have this negative charge. Now this means the DNA is open. Right? While on this side, this would likely mean that the DNA is gonna be closed, like it's gonna be tightly wound and compact. The on and off happens after this. But this is a big initial thing where I'm like, hey, you can't even touch the DNA, you're not gonna be able to turn it on or off. So that's why this is the first step of like, once it's open, now you have a shot.
That makes sense.
Just like this, let's imagine the next residue over, this is gonna be talking about phosphates, right? Or your phosphates, it's gonna be, normally I think it's for these, it's serines. What's the other one? I feel like it's serines and 309s. Yeah, well, they're the only put serines. So this will be a different amino acid. It's basically hanging out there. When you bring it down here, the modification you're gonna add, it's gonna look like this. So you're going from, you had an amino acid, and now you just made this thing super negative, right, by adding a phosphate. So in this case, what's happening is, from this going down, you have a kinase, right? It's gonna add the phosphorylation mark to the amino acid. And then the opposite one, what do we have? What's the opposite, guys?
Phosphatase.
Phosphatase, right? So they're doing their fight. So again, these molecules come in, they can modify the histone tail based on whether it's positive or negative. They'll tell us how the DNA is going to behave. Yeah. And the cool thing is, again, these two are molecules that they just do their small task. It'll be our activators and repressors that recruit them to the piece of DNA that we want. Same thing's happening here. These kinases and phosphatases, when we want them to work somewhere, we're recruiting them to that specific piece of DNA. Yeah. And the last one's going to be your methylation. So again, but it's the same principle. You're going from here, I think you have a slightly positive charge there, and down here you're adding a methyl group. But again, you know, set of enzymes, you can add the methyl group, you can take it off. So it's a way for you to switch the gene on and off by just modifying the tails. And then the only one that's involved in like, so in all these cases, I'm showing you where the tag, because of the way it's set up, the tag is being the thing that's getting us to this open DNA state, right? The only one that, by adding a tag, leads to the off state is the methylation. The only thing is like there, if you notice it, it happens at different residues. So amino acid lysine at 9 and lysine 27. This is where the modification happens here, methylation happens there, the gene's gonna actually close better. So for the most part, if it's gonna be an activation thing or open chromatid, you gotta be thinking, you can use any of the three, acetylation, phosphorylation, or methylation. And then for turning it off, it's only gonna be methylation. Like the mark being the off signal, yeah? You don't have to memorize the exact positions. I would try to memorize a little this end of the lysine. So I'll give you that. But I might give you this picture together for the test. But just be comfortable that you emphasize that positive charge, because that's the big thing. Yeah. Any other questions on this one? No.
What's this slide about? Or have we not covered it yet?
So this is kind of going down the storyline, right? So it's like, you imagine we started here, everything's tightly packed, our DNA is in this closed conformation. The activator is the one that picks the DNA site where we're gonna grab onto. It brings in, that would be the hat, right? The hat's gonna make us go from a positive charge to a negative charge. It should open up the DNA, and when it kicks off these histones, that's when our machinery to bring the polymerase can grab on and do their job.
This is like a general summary of,
Yep.
okay, gotcha.
So like I said, it's upstream of section 1 stuff. All right, so part of this, being in these different states, what we find is that in our cells, different cell types, we actually have different pieces of our chromosome already in the compact state, and we have some of them in an open state, right? So you basically open up whatever genes you need to work in those cell types. So the big problem there is that when it's time for you to replicate the DNA, you try to copy it, right? You can go from one double-stranded strand to 2 double-stranded strands. In this case, how do you pass that information to those 2 pieces of DNA, stuff that should be open, and stuff that should be closed. In this case, what they're kind of doing is they're basically opening up the DNA, having to take off the histones, and they kind of stay there locally, and then they kind of reset after they pass through. And they do this thing where they're kind of giving one histone to one histone to the other one histone to the other one, right? They're taking turns grabbing on histones, but you're creating a lot of new histones at the same time, right? So it's like if there's a lot of histones already there, when you open them up, they each are getting half of the old ones, and each one's getting half new ones to help close it up. If the DNA was open, they don't recruit that many histones, so you can leave that DNA still open on both strands. Yeah. The other way that we can mess with turning genes on and off, it's going to be with our with modifying, but this time we're going to modify the DNA itself, right? So if I do the figure here, this is your cytosine. So down here, I will draw the sugar ring, your phosphate group here, your oxygen group there. So just on the nitrogenous base, you can do these kind of covalent modifications. Yeah. I believe it was in DNA repair, I talked about this one, which is our DNA can get methylated. Old DNA will be methylated, brand new, barely made DNA will not. So when we do find an error, it knows to say like, hey, the one that looks like this is brand new, we can fix that. If it's old and it looks like this, it'll be like, no, we know this is the good copy, let's try to modify the other one. Yeah. And for this, this is the big enzyme, DNA methyltransferase. So that'll be that enzyme grabs on, and then here's, you can modify it, add that methyl group to it, right? So this one, it's like when you do the methylation event, this is going to put us into that repression state, right? So this will be the thing that turns it on, or sorry, turns the gene off. But this one is a modification directly on the DNA itself, right? But again, most of these work in complexes. Something else targets it to this gene, gets the modification, and we place it into that spot. And then this one would be the opposite reaction, right? Which is once the D is methylated, it's on its off state. You bring in this DET. This will be the enzyme that basically kind of sets off this reaction. You kind of charge it up with that hydroxyl group. Then you can try to update it with almost oxidizes to kick it off. And then you end up back here sending it back to the C state, right? So if I did my little drawing, how I did here, right? It's like we're going to the open state, going this way. You would bring in the TET. If you want to go from the open state to the closed state or turn the gene off, you bring this one, your DNA methyltransferase, right? And again, these will be the two ways that you can modify it. Turn the gene on, turn the gene off. Yeah. And that's the big stuff for today. So we had to modify the DNA, or you can modify the histones. Everyone pretty comfortable with that? Yeah? All right, any other questions? All right, write what you got in the cards if something's unclear, or propose some questions for the midterm. Okay, something like this, and then I'll take a question. Thank you, sir.
Yeah, I put an impossible question on there, but you said you weren't gonna ask it anyway, so.
I did that last time.
That's one that I was like, yeah, it seems like it might be simple, you know. What is the second part of that? DNA
Methyltransferase.
That's transference?
You know what I mean? Transferase.
Transfer. Fellas. Also, I wanted to talk about possibly taking the exam. Like Tuesday, maybe. I know you have 1:30 to 4:30 of lab, I think you said.
Upstairs. So I'll have a witness come by, pick it up, just reserve the library space now, because it takes 2 days for it to clear it.
Reserve a library space.
Yeah, I have study spots in the bottom of the library.
Can I use one of the rooms in Woods to do it? Like one of the biology rooms, or the tutor rooms to do it?
If they're open.
I don't even know what mine says.
What time do you want me to come in? Or just whatever time?
I'm gonna be there up there in the lab, and in my office. So it's like.
So anytime.
We're good.
You come in earlier, it's nicer.
Right. Alright, sounds good. Thank you.
Oh,
I know
that's
I
nice.
But
Tired, but okay.
Yeah, actually, I don't know.
oh wait, what time is it?
Okay, what do I need to do next?
Okay, but yeah.
I want to like look. I'm like, okay, what's next? Okay, what's tomorrow? And I'm like, nothing. But there is, like, I could do a lot. I should do a lot. I should sit down.
I think I'm gonna walk to the
I
library.
don't want to.
I need to do my test correction.
Yeah, I'm like, oh, never mind. And then like, you don't have to.
I need to leave it now. I'll see you guys later.
Good.
Bye.
What?
No.
No, no, studying
This?
and my other test next week that I have to read a whole book on.
I just look like a giant.
I was like, with my hair and everything, it's like I had.
Oh my gosh, okay, that's way better. My friends tell me I look like Mr. Hank.
So like a 30. Here, it's just sorting to the That's the
Did you
way
get
they have it.
Yes.
And the connections were a little not.
That could be worse.
Enhance really.
Yes.
I mean, yes, it could be a lot worse.
Transcription on it. And the enhancer, so bring this two up a little bit better.
Like distinct.
Really?
Cause how do they fit into this?
She's stressing with this.
The enhancers. This is like the initial part, right?
Who meant to
And then
read?
these come in after.
So this is, that's the DNA.
Yeah.
Right, so this one, I had almost.
Is he posted?
Storylines, right?
His lectures.
Not.
So what's this up here? This is like your color box, right? That's your DNA, and this is your, color box, proteins, like all the little machinery sets up here, the polymerase sets up there, and your mediator sets up like this.
Okay.
Then on this side, here's the enhancers are over here, which is the DNA element, and then they have proteins. And these are the proteins that then either signal here or they signal the histones.
Okay.
This and this, there, look like it's just one genome together. So split that up,
Oh, okay, that makes more sense.
because here is where you have the crazy amount of power, where I'm like, the details for the repressed side. Here, you should be saying like, hey, this one behaves one way, this over here, the correct works in X way.
Okay.
You can have multiple strategies, but once you pick one,
Okay.
show off one of those, I'll be happy.
And then one more question. Did you want me to go into more depth, like after, once it becomes a protein? I wasn't sure what this.
Good intro. Thank you, I appreciate that. Yeah. It's again there. And so these two make this the endonucleus. This is what we're cutting down. Here you find when we make maybe I'll be picky that day. So here it's like you're making the protein in the ER. Here it's yeah, so you're making it, you have XPB, and then it leaves the ER as a correctly for the protein. So now this is functional, right?
Yeah.
But what does it do once you read it?
No, it's the correctly folded protein. So then
Yes, because you had basically here, what you're saying is like there's two options, right? You either have this thing that had extra RNA, it's not a good protein, and it doesn't do anything. And you have this other one where it's like, cut out the RNA, you made a good protein, and what is it go do?
The DNA. Some measure it as.
So, yeah, but what DNA does it hit? No. All right, find
Go
out
into that.
what DNA it hits, and then you're gonna be like, oh my god,
Okay.
it's gonna like, it's gonna click. Your name, yes, that's concerned.
And then also the HIV, do you want me to do as soon as I finish?
See if you have the time, I know right now, I
Yeah.
made first number one already.
Okay.
Yeah, so get on that as much as you can, if you can study it as much as you can. The other thing you have to send me a good break. Make sure number one. But it's always good to touch base with them. Yeah, I think you have to get it done in here.
Yeah. I just have never.
You got that. I'm gonna try my best, I should be able to get, this fixed.
Okay.
All right, and then I'll let you know.
Okay, sounds good.
Yeah,
And I don't have class after.
you're free after?
Yeah.
Okay, perfect,
Thank you.
awesome.
There's my note card. At the test, like the midterm, is it gonna be cumulative, like, okay,
The goal is to avoid making it cumulative. Like,
okay,
that's why I was bringing it up today, where I'm like, hey, I want the histones.
Yeah.
To be, like this part, how they get modified, to the open and close state. Now it's open to this stuff, but I'm not gonna bring up, how does the enhancer help?
Okay,
That's just a mediator to get things
Okay.
going on.
are we going to talk about meiosis, like the X and the Y cells and stuff like that?
I don't wanna confuse people, that's why I'm trying to stay just on mitosis.
Okay, but like next semester, like next